You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. My guest today is Panna Sharma. He's the CEO of Lantern Pharma. They work in the oncology area, and I'll let Panna describe that more. So, Panna, thanks for coming.
Thank you for inviting me. I look forward to your to discussion.
Yeah, so tell me about Lantern. What's the premise of it?
Yeah, I was really intrigued by Lantern even prior to my arriving as CEO a little over a year ago. Because the company was doing really what is the next most obvious thing in the development of cancer drugs, and that is applying artificial intelligence and machine learning and leveraging all the tremendous amounts of genomic data that's now widely available to help develop new drugs and rescue drugs that have failed in oncology in the past. And oncology today, as you know, is a very personalized, increasingly more personalized science. We're understanding more about how each cancer is so different. 20 years ago, we only had 4 or 5 different known blood cancers. Today we have characterized virtually 100 different types of lymphomas, leukemias, myelomas. And they all are very different in terms of the way they're molecularly structured and in terms of what the genome of those cancer cells are. And so AI, the ability to look through all this data and to analyze data and cells and patient genomes faster and quicker and 24 hours a day, 7 days a week is a perfect problem area for AI and machine learning. And so what the premise of Lantern is to use the most current advances in machine learning, artificial intelligence, and genomics to help rescue and develop cancer drugs faster and cheaper and with lower risk so we can get them to patients in a more personalized manner. And so, we're not purely an AI company. We're not just a drug company. We're really a mixture of bringing new AI and machine learning techniques to really reinvent and accelerate the development of cancer drugs.
So why would AI be needed? What are there huge combinations or, thousands or tens of thousands or millions of permutations of certain elements of drugs?
Oh, yeah, tens of millions. Yeah, there's, in fact, could be more permutations of chemical compounds than there are, galaxies out there. So it's a perfect problem area for AI. And, just think inside of your own body, you have about, 40 trillion cells, 30 trillion of which have DNA. And every day, some of them go bizarre or errant and, your body cleans them up and it keeps disease away. But those that continue longer have the potential to become cancerous, and everyone's are slightly different. And so that's why you always hear about someone on the same therapy, whether it be a PD-L1 therapy or a targeted inhibitor or even chemotherapy. Some patients do really, really well, and they go on to have, a meaningful life, and they don't affect their families, and others, die quickly or don't respond to the drug or have other side effects. And that's not because necessarily one had a more advanced cancer. It's just these cancers are that different. And you always hear about at the same time, not only patients responding so differently, but you also hear about these trials. And, having been in the biotech space for some time, I was at another company, Cancer Genetics, prior to this public company. And we did a lot of work on cancer trials, both for biotechs and pharmas. And you'd have these phase 3 trials where the drugs were very safe and effective somewhere, but they failed because some percentage of patients responded very well, but some didn't respond at all, and others didn't quite respond the way that was predicted. But there were patients that really got a lot of value, even though the drug failed, because it wasn't effective enough, or it wasn't more effective than the standard of care. And those patients, though, there were probably other patients like that, and they were not going to get the benefit of having that drug on the market. And now that we have precision pathways and we understand why those patients responded really well, and we can predict that with machine learning and AI, we can bring those drugs back to market. And so that's also, to me, is a wonderful way to accelerate the delivery of shareholder value. You have these drugs where hundreds of millions of dollars have been put into the development of that molecule, and then it gets shelved or abandoned, not because it wasn't good, but because it wasn't good enough at the time that we were doing trials. And today, the world of clinical trials has totally changed.
What is good enough, by the way? Is it just expressed as a percentage, or like what are some of the factors that tell you?
You have to have a discussion. Yeah, you have to typically have a discussion with the FDA, but typically that is better than the standard of care. Notable improvement over the standard of care. If it's a rare disease or an orphan disease where there is no cure or where there is nothing that's really used, then you can have maybe criteria that aren't as strict. But typically in cancer, it has to be statistically more effective and more safe, measured by safety, than the existing standard of care. But a lot of these cancers that we're discovering, as we discover more and more subtypes of cancer, and I'll give you some examples from our own portfolio of drugs, there are no cures. And so the ability to bring these drugs into the market for some of these subtypes of cancers is really a phenomenal opportunity if we can really show that we can determine which patients will respond and why they'll respond. And again, these are now problems that are central to biology and discovery. And 5 years ago, we couldn't spin up 100 supercomputers in the cloud for a few $1,000s and manage it. And today we can. And that's a phenomenal change. And so rather than applying it to making cars or to predicting weather or predicting traffic patterns or sending Uber drivers to your home, we're using those same advances in machine learning and AI and data manipulation and cloud infrastructure to solve the problems around cancer and cancer biology.
So just as an example, are there any particular cancers where the numbers on the standard of care, like what percentage cure rate there is, or is it expressed in months the person lives?
There are some of those. I can give you some examples. Specifically, I think probably from our own portfolio would be best. Our most advanced, I'll give you an example of LP100, which is Lantern Pharma 100, our most advanced drug that's currently in a phase 2 trial that's actively enrolling in Europe with our partner. And we rescued that drug because our founders and chief scientific officer and prior management team, before I got there, felt that drug had really good phase 2 data, which it did, but then it failed phase 3 because it was tried in a prostate cancer indication. And it worked in about a little less than 15% of men, which wasn't deemed statistically good enough. So, only 15% of the population in prostate cancer it worked in, and this is almost 10 years ago, they deemed that was not going to be effective enough. But in that 15%, or a little bit less than that 15% that it worked in, it worked extraordinarily well. And so there was a reason for that, and biologists at the time knew that there are different types of prostate cancer, some that you could have for 20 or 30 years and not die from, and some that you could die within 2 or 3. But they didn't fully understand the mechanisms. Well, fast forward to 2017 and 18 and 19, and now we understand the genetics of prostate cancer much more clearly. We understand the ones that are more aggressive, the ones that are less aggressive, the ones that respond to hormone therapy, the ones that have to be removed. And so the science in cancer biology is advanced. And so we can go back and say, well, this drug worked in this subset of patients. Why was that? And so the hypothesis that we had is that it worked really well in patients that had metastatic or aggressive disease because of basically because of DNA damage, repair, and inhibition. Anyway, so we rescued that drug by looking at that thesis. We looked at, okay, well, this trial should be redesigned for men who have prostate cancer where it's now metastatic and where they are hormone refractory. And again, that's about 12% of the population globally gets that disease, 12% of the prostate cancer population. So that's a subset of prostate cancer. And so with that thesis, we were able to convince the regulatory bodies that, look, here's the underlying data on why this drug works in this subset because of this mechanism. And this mechanism is only mostly involved in these patients, and our drug works actively here because of these reasons. And they said, well, great, let's design a trial around that subset of hormone refractory metastatic prostate cancer patients. And that's what we're doing. So it's a very targeted population. And so far, we're about halfway through the trial, and the patients are responding well, and there's been a high level of tolerability of the drug. So we're very hopeful. And again, a lot of these paths didn't exist, 5 years ago.
Where does the AI come in? What requires the intense computation?
Going through thousands and thousands of genes across hundreds of samples or hundreds of different scenarios. So it's, we're looking at a lot of biological filtering and correlation analysis and, trying to do it in tandem, because as we enroll these patients have to be not only metastatic, which is easy to tell, but they also have to have a certain gene profile. And so affirming that gene profile takes time. It could take historically, when I was in my prior biotech, it would take us maybe 2 years to develop a good genetic signature. And that was if we had some good ideas about the genes that were involved and, the pathways. And it would take an interdisciplinary team. Not that you couldn't do it without AI, but you could. But it would take a good 2 years. And, a good cross-functional team, a lot of really smart people working together, biologists, geneticists, bioinformaticians, lab folks. And so it was a complex and expensive process. And so that process to develop that genetic signature now can take, instead of 2 years and a team of 10 to 12, it can take us maybe 3 months and a team of 2 or 3. And that's because we're able to put this data in the cloud and crunch through the millions and millions of permutations of genetic signatures, do back testing, do various forms of analysis, put scenarios in place, and get more confidence that the signature we're coming up with is going to be predictive of the outcome that we want. And so AI really allows us to do it cheaper, faster, and with greater confidence.
So what do you envision in the future as to, the role of, do you think AI will allow N of one or just literally individual customized treatments for people? Like you'll be able to sequence and profile someone and come up with a drug scheme that's literally unique to them, whether it's the strength or combinations, whatever.
Yeah, or even combination therapy. So as you come up with combination therapies, more than one drug being used at a time, like it is, for example, in AIDS, it's a combination therapy ultimately that has kept AIDS at bay and has worked really well. It's, 3 drugs being worked on together. Eventually in cancer, I think a lot of the cancers will not necessarily go away, but they will be better managed, and they'll be managed through combinations. And those combinations, I think, are going to become increasingly more and more personalized. Maybe not N of 1, but maybe it's, N of 1,000 or N of 500 or N of 50. But yes, there will be increasing levels of personalization in cancer therapy. And this, just purely just the numbers. If you look at the numbers of all the genetic data that we're gathering on patients, both pre-cancer as well as after they get cancer, all that data needs to be put to good use. And, you just can't, fill up rooms and rooms full of just, bioinformaticians and statistical folks to do that. They have to eat, they have to sleep, they have biases. And so this is, again, a perfect problem where you can develop AI, and that AI can go on to develop its own AI. So, instead of just using one algorithm, you can actually have it perform and create other algorithms. So that's also the very interesting thing around where AI is going. So as you do kind of the, nearest fit and nearest neighbor, and you create different algorithms and train them, every time you do one of these sets, you learn something new. And so that is, one of my visions is to really create a process where we're constantly refining and updating the algorithms by which we're identifying how drugs interact with our genes, and more importantly, are able to predict how those genes are going to allow us to either respond really well to a drug or not, or to a drug combination. And so I think that is the future of how drugs will be developed, and more importantly, how trials will be designed. And so that's, I'm really excited about, because that's a multi-billion, trillion-dollar problem. And right now, as you know, it costs almost $2 billion. If you look at different studies, anywhere between $800 million and $2 billion to develop a cancer drug. And we need to take some numbers off that. We need to take some zeros off that, especially given how expensive cancer therapy has become. And so if you imagine that our population is only going to get older and cancer will become more prevalent as we age, how do we continue finding solutions? And so part of that is I believe, is definitely going to be the application of machine learning and the application of high throughput genomic analysis to develop and deliver these therapies.
So have you seen any commonalities that can make your models more efficient? Because you're using AI, maybe in the solution space or the answer space, you're seeing areas that can be cut out or assumed or baked into the model to make the models themselves, exponentially more efficient.
Not yet. I don't think we've exhausted enough of our space. Right now, we have about 100 million data points, close to 100 million data points, real-world data points of drug-tumor interactions in our model, radar, in our platform. We'd like to get to about 350 million next year. And right now, again, our model has about 140 different drugs built into it. And again, I'd like to increase that number a little bit also. So I think there's no real, I would say, commonality. There are certain things that we're seeing inside of certain cancers, like prostate, for example, or non-small cell lung cancer. There are certain ways that we're tweaking the algorithm based on our understanding of what goes on in that cancer. So we give certain biological filters more weight than others based on our discussions with experts. And so, some of that, I would say, maybe not the models themselves, maybe certain weights or certain traits or certain filters we give a little bit more weight to, or a little bit more bias towards when looking at a specific subtype of cancer. If that makes sense to you.
Yeah, I just wondered, because again, the solution space is so vast. It would be nice if AI found a way again where you can lop off maybe a tenth of the solution space, because it seems to have commonality, or maybe that would elucidate common pathways to attack any kind of cancer. I don't know. I guess one way is to solve it for the individual person, and one way is to find commonalities that would apply across, a vast number of people.
No, yeah. I think we are. There are certain, like I said, there are certain filters that we use. So, for example, if we find someone that has we're working on a certain problem right now in breast and ovarian cancer, and we are making certain assumptions if people have BRCA1 or 2 gene mutations or certain germline mutations that they inherit. And so we are making certain assumptions there about how that cancer may respond to one of our drugs versus if patients don't have it. And that is definitely something that we have learned, and also, more importantly, proven through the literature. And also, we've modeled out in cell line and rat studies as well. So, I would say that we're finding those, although this problem area is so new that we are very, very slow to make biases or make certain shortcuts or conclusions, because we're just learning so much more every month right now.
I gotcha. Are you profiling then the constituent cells of tumors in people that have various cancers? Are you looking at?
Yes, we are.
Okay, so you're looking at the gene expression of those cells, even though they're heterogeneous. You're able to look at, how many different types of constituent cells of a given cancer, a given tumor?
It depends the number, but typically we try to get a pretty high tumor burden in the cells we look at. So even this, so we are, when we get, for example, a biopsy of ovarian or liver or prostate tissue, we're trying to take only the cells that have and that are cancerous based on the pathology report or based on the tissue analysis. And so the tumor burden in the cells that we're looking at is fairly high. So it's not like, it's 1 out of 1,000. We're looking at tumor burden that's often, 10, 15, 30, 50%.
Well, okay. But even within there, let's say if you're just looking at a solitary tumor, from what I've heard, in order to identify it, it has typically a billion cells in it. How many different types of, not really cell types, but how many different gene expression profiles are there within a given tumor even? Have you guys elucidated? There are thousands, millions?
There definitely can be hundreds. And there's teasing out that commonality as part of the challenge. Absolutely.
Has there been much observed commonality in their expression, or is each expression pathway like totally different?
So within a tumor, there is some observed commonality, and that's in the literature, and so we try to work off of that. But also the expression patterns will change based on how the drug reacts to the tumor, and so that's also a big part of our analysis and secret sauce. Is we don't really care just about the underlying tumor. What we also care about is what is the tumor's response to our drug or to our drug combination. Because eventually our goal isn't just to diagnose the patient at all. Our goal really is to treat the cancer with our drug. And so we're really focused on what is the effect on that expression level. Even if the expressions are different, does it have the desired effect to kill off the cell? And do we see a sign of that once the drug is eluted into the tumor or into the cell line or into the sample? So one of the studies that we're doing right now with one of our partners, Center for Translational Research and Investigation in Northern Ireland, is that we're taking live prostate biopsy material from patients who are already going to undergo, traditional prostate cancer therapy, which is typically radiotherapy or ablation or removal. So they're going to undergo that. And so we get a piece of the prostate biopsy. We then expose that fresh biopsy material to our drugs, both as monotherapy and also as combination therapy, to see if it has the effect of killing off or, reducing the tumor burden in each of those wells. And so that gives us a sense of, what is the right drug to potentially give to these patients if the prostate cancer recurs. And so, yes, the initial starting expression level isn't going to be the same, but there will be similarities in patterns. But then the key pattern we're looking for is, what is the expression level once our drug is introduced into the tumor system? Are we seeing enough of a signal, and can we predict where it'll be effective and where it will not be effective?
Are you using multiple drugs or just one drug at a time?
We're testing for both. So we have our own drug. We have some various forms of our one drug. So we might have a positive enantiomer. We might have a negative enantiomer. We may have a mixture of both, what's called a racemic mixture. We may have our drug with the existing standard of care, our drug with a chemotherapy agent, our drug with another potential novel drug. So we look at various forms to really optimize, how is our drug going to work best. And we also, sometimes depending on the study, we may have it at different, dose ranges to do dose range finding. And again, a lot of this work isn't just done in the lab, but it's also done in silico. We try to do this work in the machine also so that we can check our predictions in the machine versus reality. And we use whatever deltas or differences that we learn to make the next iteration of predictions better and better. So it's, really, I think this is kind of the future of drug discovery, where you're using machine methods alongside traditional laboratory methods. And, our team is a perfect example where, half of them are traditional cancer biologists and cancer researchers, and the other half are AI and bioinformaticians and machine learning people. And so it's a real mixture. And I think that really is the future of how drug discovery will become more efficient and more productive.
Are there, so you're looking at the DNA level. Are you looking at are you able to look at epigenetic?
We're mostly looking at the RNA. So we're looking at RNA, which is expression level, and we do look at DNA. Again, DNA will give us a good indicator of what the disease is or subtypes, but DNA doesn't do a great job of telling you necessarily what the drug interaction will be inside of the cancer cell. And so that's why we look at RNA expression, or sometimes we even look at epigenetic expression.
Well, that's what I was going to ask you, because you look at RNA expression, you're probably capturing some, at least, of the epigenetic changes that occur.
Yeah, we are capturing it. And sometimes, like you said, in some cancers, it's more useful and known that, these epigenetic factors will drive drug reaction or not drive drug reaction. So again, it typically depends on the tumor subtype.
Are you able to look at and see if there's a lineage in a given tumor, meaning which cells came first, which led to other cells? It's very heterogeneous, but legend has it, tumors start out as one cell, and then they they divide and grow into billions of cells. So are you able to trace the path of growth?
We're probably not able to trace the path of growth, but what the question you're referring to is a bigger one, and that's around clonality. And that we are able to do, although in some of our studies it's not that important. But clonality is important because a tumor may arise not from just one cell, maybe a number of cells that are cancerous. And those clones, those initial cells, can be very different, and they oftentimes fight with one another. And so in many cancers where the cancer will come back, lung cancer being one of them, multiple myeloma being another one, brain tumors being another one, oftentimes you'll treat the initial clone cancer that basically has outgrown the other cancers trying to compete. And as you continue treating that initial clone and the cancer cells related to that clone will die off because you've weakened them or you've compromised them, and allows the cancer cells from another clone to take over. And oftentimes you're still working on killing the initial clone, and it gives weeks or months for the other clone to take over. And you often hear of someone responding really well to treatment, and then 2 years later or a year later or some time later, they'll die again of cancer. And that's not because that cancer came back. It oftentimes is that it was a different clone in that same cancer that took over. That happens in multiple myeloma quite rapidly. And so that's one of the areas where I initially got a lot of knowledge about, these clones and how these clones would compete for taking over the body, and then lung cancer. And so again, my prior job, I did a lot of work with the Mayo Clinic, and in multiple myeloma, we had a partnership, a joint venture, and where we got to learn a lot about the clonal theories around cancer. And the same thing in lung cancer. Our company, we did all of the testing for one of the biggest lung cancer trials, an international trial, where massive amounts of biomarker work. And again, we saw that the analysis of the initial DNA and RNA and some of these protein levels weren't sufficient enough to really correlate to response until you started looking at things like expression and other epigenetic events. And again, there were some theories that oftentimes, you would treat one clone, and that's why someone would come back 18 months or 14 months later, because that was just enough time for the other cancer to grow and take over. And so although that isn't part of our study today, again, we're not really a diagnostic company, we're a therapy company. It does help advance our understanding of cancer, and it does help us advance perhaps that we should look at combinations, especially if we know that there are multiple clones at work. If we can diagnose that earlier, perhaps we can also then improve the outcome by suggesting a certain therapy earlier. And so that's something that, yeah.
Because I had thought the theory was that, let's say you have a cancer and you hit it with chemotherapy, the cancer cells themselves change and their gene expression changes, and then they're able to resist that treatment in the future. But you're saying there's at least the secondary, maybe the predominant mechanism is that it just allows other clonal lineages to compete more effectively, and thereby those arise and they wouldn't respond in the same way as the previous lineage.
Yeah, both things are happening. The cells are exchanging their expression because they are becoming more resistant, they're becoming smarter. But oftentimes, the other thing will happen is that, some of these cytotoxic or chemotherapies are so powerful, they kill off so many cells, both wanted and unwanted, and it allows a totally different clone to arise. And that definitely is what happens in certain cancer types that have been modeled more closely, like multiple myeloma, like certain B-cell cancers, like some certain lung cancers as well. But both things are at work that you mentioned. One is, yes, they do become resistant and they become smarter, but at the same time, there are typically in some of these cancer categories, there are multiple clones that are trying to compete.
Yeah, that's really interesting. I didn't know that. It reminds me of like the microbiome. If you have a healthy microbiome, then you take antibiotics, for instance, and they kill off the predominant actors and allow things like C. difficile to take over, which is, let's say a pathogen. It sounds like in cancer, maybe something similar. I'm just speculating, but you have a certain cancer that acts, and then you attack it with chemotherapy, it retreats, but then it allows a more aggressive cell to predominate and take over.
Yeah. So again, modeling this through is exactly why, having a data and AI-driven platform helps. It allows you to ask questions faster. Not necessarily that you'll be right it's just, it allows you to ask and iterate more rapidly. And with that, you can identify, potentially more drugs and more drug types, and perhaps even, develop things so that you feel more comfortable taking them into the clinical setting. So again, for us, AI it is not the answer. We're not an AI company. We're leveraging AI as the latest tool to basically develop drugs faster, cheaper, better, and more targeted. And so it's, that's why, again, we continue to think AI is a perfect type of technology and approach for drug discovery, and specifically in cancer, because we have so much genomic data and so much more biological data available to us.
Do you think there's a fear of complexity that holds back, scientific effort? Because I've spoken to many, many companies, and I'll say, what about this? What about that? And they'll say, oh, well, we focus on this. You think it's because shying away from the true complexity of the thing? And, because you use AI, does that allow you to embrace it more and incorporate more into your models?
It definitely there is, because, machines just have greater hard memory and don't eat or sleep. There is a way that we can start asking bigger, broader sets of questions. We can look at bigger problem areas. But like anything, I think you have to really focus if you're going to solve a problem. So there are reasons why we're not looking at Parkinson's or Alzheimer's or multiple sclerosis, all of which have, their own sets of data and their own sets of issues, and, probably can benefit from machine learning and AI-driven approaches. So I think there is something to be said about focusing. So we're focusing initially only in small molecules, mostly in solid tumor, where we can derive a clear stratification for response. And so that's our first initial area. Doesn't mean that we won't expand beyond that. But I think once we prove to investors and more, to patients and investors, we can do that well, I think then we can ask bigger problems. So we can, as you mentioned, go after vaster areas of the problem area. But yes, machine learning definitely allows you to do that. There has been no database that I know of, or no platform that's trying to approach reaching, 1 billion data points of drug-tumor interactions specifically in oncology. And so that's where we will be by the end of 2021. And so I think, that just allows us to ask and look at questions in a totally different level than has been done before in drug development.
Do you have a preferred model or substrate that you use? Like, do you work with companies that make organoids? And is that your preferred model to test the efficacy of drugs, or is there another way?
I think one of the things I always learned early on in cancer is you cannot have preferences. Every cancer, with brain cancer, liver cancer, stomach cancer, they're all they all can be very, very different. But yeah, we use 3D models like organoids. In fact, we have a collaboration we'll be announcing with a major university and medical center where we're looking at specific types of organoids to augment our data in urogenital cancers. But yeah, we do think the use of 3D models and organoids, if done properly and with the right quality control, the right background on the organoids, analysis of the lineage of the initial cell lines, that they can be a very, very powerful tool and accelerate studies. But like anything, you got to be very thoughtful about how the organoids were developed and what the precursor cell was and what the process was and what genomic drift happened between taking it from the patient to the actual organoid being developed. And so those are all things that you have to QC for.
Well, very good. Well, Panna, this has been super interesting. What's the best way for people to find out more about what Lantern's doing?
Yeah, obviously our website, Lantern Pharma, is a great place. We have three drugs in development, another early stage compound that we're refining, and we have active clinical trials going on. But lanternpharma.com. They can also follow us at LinkedIn or on Twitter at Lantern Pharma. And I often talk a lot about AI and cancer at my Twitter handle, The Real Panna, Panna. And so they can also learn a lot about kind of the leading edge issues around genomics and AI around cancer there as well.
That's great. Well, Panna, thank you so much for coming.
Thank you very much for your time. I appreciate the questions and I love the conversation.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, where we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.